So very good afternoon to all of you. Today we are going to see the major part that is associated with ELISA. And this ELISA, in my previous lecture, we have gone some basic part. But today we'll try to continue with the components, the specificity of ELISA. So without wasting much time, we'll go with ELISA. Now, what this ELISA will consider of? It will be consist of ELISA plates, buffers, antibodies, enzymes, sample handling preparation, and substrate. So all these components are to be there, or they have to be there in order to make a proper ELISA technique run. ELISA plates. So what are these ELISA plates are up to? Plates, flat bottom 96 well plates made from polystyrene or polyvinyl chloride are coated with either inactivated antigens or antibodies. You can see in this picture. Here, those particular plates are there, and these plates will be helpful for the. So these plates are there. You can see here, these are the plates itself, and these plates will be helpful. Sorry, these plates will be helpful for identification. Now, these plates are been coated with the help of inactigen, either the antigen or the antibody are being coated inside this particular well. The coating acts as a binding site for the antigen and antibody present in the sample. Further variants of 384 wells and 1536 well plates are there. So the size of the well plates is very high. You can easily go along with it. Now, what are the plate characteristics? Its main consistency minimizes edges effect and provides optimal conditions for data collection. So it is helpful for a clear visualization. Low to medium type, very small amount of sample is being needed when you put that sample inside the well. Therefore, these wells are said to be well being prepared. Antigen and antibody pre-coated plates are also commercially available. Of course, these are now commercially available. Enzymes like the industries like Pfizer or Sigma, they produces this particular kind of plates and which are commercially now been available. Now buffers, standard buffers. Several different buffers are used during an ELISA, one for coating, another for blocking, another for washing, and perhaps another for sample and antibody dilution. So there are a number of buffers that have been utilized because since this is an enzyme-based reactions, buffer plays a very vital role because if somehow if the pH fluctuation is there, then the enzyme will not work in a proper way. It will not work out on its substrate, and ultimately you cannot get a proper result. Therefore, the buffer preparation has to be very perfect. Buffers can be produced in-house or sourced from a variety of commercial antibody and reagent supply. Of course, in a kit of ELISA, the buffers are easily been available, which are being manufactured by different companies, and they are being helpful for analysis through ELISA. Coating buffers. Coating is the process where suitably diluted antigen or antibody is incubated until absorbed to the surface of the well. Absorption occurs passively as the result of hydrophobic interactions between the amino acid side chain on antibody or antigen used for coating and the plastic surface. It is dependent upon time, temperature, and pH of the coating buffer as well as the concentration of the coating reagent. So, in order to coat any antigen and antibody inside the well, the coating buffer is said to be an important part. Typical coating conditions involve 50 to 100 microliter of coating buffer containing antigen or antibody at a concentration of 1 to 10 microgram per ml and incubating overnight at 4 degrees centigrade or 1 to 3 hours at 37 days. So this is a procedure by which the coating of a particular antigen inside the well can be done. So coating buffer stabilizes the antigen and antibody which is used to coat the ELISA multi-well plate, maximizing absorption to the plate and optimizing interactions with the detection of the antibody. The two most important coating buffers are bicarbonate buffers at pH 9.6 and PBS are there which are helpful for coating. Blocking buffer. To prevent the non-specific binding of detection of antibodies, there are two main types of blocking reagents, proteins and detergents. Protein permanent blocking agents and hence added after the capture antibody is absorbed to the well surface. Detergent blocking temporarily blocking functions disappearing during washing steps. The most basic blocking buffer contains 1% BSA or milk proteins dissolved in PBS. Usually 150 microliter of blocking buffer is used to the well to incubate for 1 hour at 37 degrees centigrade in order to fully block the flame. So blocking buffer are also useful for ELISA technique. Washing buffers. To remove the unbounded material, usually PBS with a small concentration of non-ionic detergent such as Queen 20 is used. Washing is typically repeated 3 to 5 times. Between each step in the ELISA, thoroughly remove unbound material. So unbound material is being removed because of this. Excess wash solution must be removed in the final wash step to prevent the dilation of the fragments added in the subsequent stages. ELISA antibody. Now this is important. This is a very important part. The antibody used in ELISA may be a monoclonal, polyclonal, or combination of both. So why monoclonal? Monoclonal means it has a single specific epitope, so that a specific antigen, when you put inside it, it will bind to that particular antigen or antibody interaction would take place in a very specific manner. The interaction between antibodies and their antigen is described in 3 ways: specificity, affinity, and avidity. What I told you just now. Specificity is an indication whether an antibody binds solely to the unique antigen. So there should be a uniqueness between the antigen and antibody. Affinity describes the strength of the binding of an antibody to a single antigen, and avidity accounts for the total stability of the antibody-antigen interaction. So it has to be stable also. Means it should not happen that when an antibody is there, an antigen is binding to it, and again it is dislocating from each other. This should not happen. So all these things need to be considered in that way as well. Monoclonal and polyclonal, both types of antibody can be utilized into this context. Enzymes, this is important. The most commonly used enzyme labels are horseradish peroxidase, HRP, alkaline phosphatases, AP, beta-galactosidase, acetylcholine transferase, and catalase. You see, all these are enzymes, and once these enzymes are there, yes, you need only to drop the substrate inside it, which is having a chromophore, and ultimately the change in color can be detected. The specimen sample for ELISA includes serum, CSF, sputum, urine, semen, supernatant of culture, and stool. So all these samples can be analyzed with the help of technique of ELISA. In the simplest case, ELISA samples are evaluated in PBS, washed buffer, or other specialty buffers and applied in the final volume of 100 microliters. So very small amount of volume has been required for the analysis, and these are the list of samples that can be utilized for ELISA purposes. Substrate. Large selection of substrate is available for performing ELISA with HRP and AP conjugate. HRP, ABDS, that 2,2-azinobis, 3-ethyl benzothiazole, 6-sulfonic acid, diammonium salt, OPD, TMM. With alkaline phosphatases, it will be PNPP, that is para-nitrophenyl phosphates or disodium salts. The choice of substrate depends upon required assay sensitivity and the instrument available for signal detection, spectrophotometer or fluorometry. So these enzymes have specificity towards its substrate, and these substrate can be utilized, which once been inside that plate will get converted into product, which we can analyze spectrophotometrically or with the help of a fluorometer. So that's all for today. These are all the components that we have seen, and these components are said to be helpful for the proper detection of a particular antigen present in the list that we have seen. That list should contain, it may be urine, it may be a semen sample, it may be a blood sample as well for detection of that particular antigen.